Good morning students and welcome to video lecture on Biomolecules-III, that is enzymes. All the biological reactions are catalyzed by biocatalyst in living organism called as enzymes. All the biological or biocatalyst which catalyze the reactions in living organism are called enzyme, and chemically all enzymes are proteins and they are required in very small quantity as they are catalysts. Also they reduce the activation energy for a particular reaction. For example, enzyme maltase converts maltose into glucose. So on page number 314 there are some questions. Which parameters, equilibrium constant or activation energy, decide the rate of chemical reaction? So the activation energy of chemical reaction is closely related to rate. Higher the activation energy, slower the chemical reaction. And second question is, what is the influence of catalyst on activation energy? Catalyst increases the rate of chemical reaction by lowering the activation energy so that more reactant molecules collide with enough energy to surmount the smaller energy barrier. A very large number of chemical reaction takes place in our body. These are brought about by physiological pH of 7.5 and body temperature 37 degrees Celsius with the help of biological catalyst. For example, insulin, an enzyme secreted by pancreas, control blood sugar level. Enzymes or proteins act as biological catalyst. The molecules upon which enzyme act called substrate, and enzyme converts substrate into different molecules known as products. Now mechanism of enzyme action. Action of enzyme on substrate is described as lock and key mechanism. Accordingly, the enzyme has active sites on its surface. A substrate molecule can attach to this active site only if it has right size and shape. Once in the active site, the substrate is held in the correct orientation to react and form the product of reaction. The product leaves the active site and enzyme is then ready to act as a catalyst again. Formation of enzyme substrate complex has very low activation energy. That is how the rate of chemical reaction is very high. Some enzymes are so efficient that one enzyme molecule can catalyze a reaction of 1000 substrate molecule in 1 second. Now industrial applications of enzyme catalyst. First, conversion of glucose to sweet tasting fructose using glucose isomerase. Manufacture of new antibiotic using penicillin G acylase. Manufacture of laundry detergent using protease, and manufacture of esters using cosmetics using genetic engineered enzymes. Now next is nucleic acid. There are some questions. What is the simple term that answers all the following questions? What decides whether you are blue-eyed or brown-eyed? Nucleic acid, DNA. Why does wheat grain germinate to produce wheat plant and not rice plant? Nucleic acid, DNA. Which acid molecules are present in the nuclei of living cell? Nucleic acid, DNA plus RNA. Now nucleic acid. Nucleic acids are unbranched polymers of repeating monomer nucleotide. In other words, nucleic acids have polynucleotide structure which in turn consists of base, pentose sugar and phosphate moiety. So the nucleic acids are the biomolecules which are found in nuclei of living cells in the form of nucleoprotein chromosomes. Now, what is meant by nucleosides? Nucleosides contain two basic components of nucleic acid, that is pentose sugar and nitrogenous base. A nucleoside is formed when one position of pyrimidine or 9 position of guanine or adenine base is attached to C1 of sugar by beta linkage. And nucleotide consists of three basic components of nucleic acid, that is pentose sugar, phosphoric acid and nitrogenous base. These are obtained by esterification of carbon OH group of pentose sugar by phosphoric acid, and nucleotides are joined together through phosphate ester linkage. Then this slide shows nucleotide and nucleoside difference. Then primary structure of nucleic acid. The primary structure of nucleic acid is a sequence of nucleotide in it. This in turn determines the identity of bases in the nucleotide. Different nucleic acids have distinct primary structure, and it is the sequence of bases in DNA which carries genetic information of the organism. यहाँ पे स्ट्रक्चर दिखाई है न्यूक्लिक एसिड की, ठीक है. सो इन दिस वीडियो लेक्चर we have studied about enzymes, nucleic acid, nucleotide, nucleoside and enzyme catalyzed reaction. Thank you.